You know, you're never really alone. I mean, right now, there are literally trillions of microbes living inside you. It's a whole invisible universe that actually outnumbers your own cells. This is your microbiome, and for the longest time, it's been a total mystery. But today, we're going to pull back the curtain and see how scientists are finally starting to decode it. But think about that for a second. How on earth do you study something that complex? It's kind of like trying to map an entire rainforest by looking at just a single scoop of soil. We're talking about thousands of different species, all interacting in this super complicated dance. So, how do scientists turn all of that chaos into a clear picture of our inner world?
Well, the answer isn't a bigger microscope. It's much, much more powerful software. See, scientists now have this incredible digital toolbox, a whole suite of amazing algorithms built specifically to make sense of this beautiful biological mess. So, come on, let's open up this toolbox and see exactly how it works.
All right, first up, our inner universe. Before we jump into the how, let's talk about the why. Why does any of this even matter? Well, it's because your microbiome isn't just some random collection of germs. It's way more than that. Think of it like a bustling city inside you, a living, breathing ecosystem that is absolutely critical for your health.
And what's wild is that every single person's microbiome is as unique as their fingerprint. But when this delicate ecosystem gets thrown out of whack, a state scientists call dysbiosis, it can have some pretty serious consequences. We're seeing links to everything from Crohn's disease to obesity. So the bottom line is simple. If we want to understand our health, we have to understand this community.
So how do scientists even get their first peek inside this world? Well, it all kicks off with DNA. But the raw data they get back is, to put it lightly, a complete and utter mess. And that brings us to the first huge challenge: assembling the genetic puzzle.
Okay, I want you to imagine taking 1,000 different jigsaw puzzles, chopping up all the pieces, and then just dumping them all into one giant box. That's pretty much what scientists get from a metagenomic sample, this chaotic jumble of short DNA fragments from all the different microbes living together. Their first job? Sort this whole thing out. So, they tackle this puzzle in 3 big steps. First, they've got to clean up the data. They use a tool called Bowtie 2, which basically acts like a bouncer at a club, kicking out all of your human DNA because it doesn't belong there. Step 2 is assembly. This is where an algorithm like MegaHit looks at all those tiny overlapping DNA fragments and digitally stitches them back together into longer, more readable chains. And finally, step 3 is binning. This is where they sort all those assembled chains into, like, digital buckets, with each bucket representing the complete genome of a single microbe.
And the result of all this digital wizardry is something truly revolutionary: a metagenome assembled genome, or what they call a MAG. Just think about this for a second. It's the complete genetic blueprint of a microbe, but it was reconstructed entirely by a computer. No petri dish, no growing things in a lab. This is a total game changer because it finally lets us study the thousands of mysterious microbes that just refuse to be grown in a lab.
Now, not all these reconstructed puzzles turn out perfectly. The quality of the final genome really depends on 2 main things. If a microbe is super common in the sample, meaning you have tons of its puzzle pieces, you get a beautiful, complete picture, like you see on the left. But if a microbe is really rare and has a huge, complicated genome, well, you might only end up with a few scattered pieces, making it way harder to see the full picture.
Okay, so we've figured out who is living in our inner universe. But that's only half the story, right? Now we get to the really exciting part. What are these microbes actually doing down there?
A genome is basically a microbe's instruction manual. By reading its genes, we can figure out everything it's capable of. This whole process of figuring out what each gene does is called functional annotation.
And to do this, scientists kind of act like detectives. They take the genes they found and run them through these massive public databases, where each one answers a different question. They can check a database like KZ to ask, hey, can this microbe help digest the broccoli I had for lunch? Or they can use a tool called anti-smash to see if it's maybe secretly producing a powerful new antibiotic. It's like building a complete resume for every single microbe in the community.
And this is where it gets really, really powerful. In one study on type 2 diabetes, researchers used these exact tools. What they found was that putting patients on a high-fiber diet didn't just change who was there, it changed what they were doing. The entire microbial community retooled itself to become a factory for breaking down starches and making all these beneficial compounds. It turns out your diet can literally reprogram your gut. How cool is that?
So, we know who's there, and we know what they can do.
But here's the thing. Microbes don't live in a bubble. So the next step is to understand how they all interact with each other, to map the entire microbial social network.
Because that's what a microbiome really is, isn't it? It's not just a list of residents. It's a bustling city with a super complex social structure. You've got friends, rivals, collaborators, and competitors, all shaping the overall health of the entire ecosystem. And at a really basic level, these relationships are either positive or negative. You've got the positive ones where two microbes help each other out. When one thrives, its partner thrives too. And then you've got the negative relationships where they're both competing for the same food. In that case, if one's population goes up, the other's is forced down.
So how do scientists figure this stuff out? Well, they look for patterns across hundreds of different samples. If two microbes are almost always found together in high numbers, that's a strong positive link, and it gets a score close to +1. But if one always seems to be missing when the other one is around, that's a strong negative link, scored near -1. By doing this for every possible pair, they can build a stunningly detailed map of the entire social network.
Okay, this brings us to the final and, honestly, the most powerful part of the toolbox. Now that we have all this information, who's there, what they're doing, and how they're all connected, can we actually use it to predict health and disease?
The very first step is just to see if there's a difference at all. A statistical test called PermaNova basically asks a simple question. If you look at the entire microbiome community of a healthy group and a sick group, do they look fundamentally different? It's kind of like asking if the overall mix of trees in a rainforest is different from the mix of trees in a pine forest. You're just looking for that big picture signal.
But just knowing that the healthy and sick forests are different, that's not really enough. The real goal here is prediction. Can we look at a single person's microbiome and predict their risk for a disease, or maybe how they'll respond to a certain diet? To find those kinds of subtle, predictive patterns, scientists need to pull out the biggest gun in their toolbox: machine learning.
One of the most popular methods is called a random forest. And yeah, it's pretty much what it sounds like. The algorithm builds thousands of different decision trees, like the simple one you see here for predicting IBD. Each tree gets a vote on whether a person is healthy or sick. By combining the wisdom of thousands of these votes, the model can uncover incredibly complex patterns that link specific microbes directly to a health condition.
And this is where it gets amazing. The models don't just make predictions, they tell you why. I mean, just look at this. On the left, a model was trained to predict how healthy a person's diet is based only on their gut microbes. But then, and this is the incredible part, on the right, it spits out a list of the exact microbes that are the strongest indicators of a healthy diet and which ones are linked to an unhealthy one. It's not a black box, it's a discovery engine.
And all of this hard work leads to real-world breakthroughs. By applying this entire digital toolbox, scientists have discovered amazing things, like a totally new probiotic, a unique strain of Bifidobacterium longum that has these special enzymes to digest both milk and plant fibers. That's a truly powerful little microbe.
So you go from a chaotic soup of DNA all the way to a specific, actionable discovery. That is the incredible power of this computational toolbox. And it leaves us with a fascinating final question. As these tools get even more powerful, how long will it be before a doctor analyzes your unique inner universe and prescribes not just a medicine, but a personalized set of microbes to optimize your health? The future is coming, and it's microscopic.